Okay, I wanted to introduce you guys to the concept of G-protein coupled receptor signaling. This is something that is very common in the body, from special senses, smell, taste, even sight, to how hormones work, epinephrine, norepinephrine, the adrenaline rush you get. It's all G-protein coupled receptors. These are very, very common, and we just wanted to give you a couple of examples of how these work. The receptor itself is made up of, well, the receptor is a 7-pass transmembrane receptor. So we'll draw it like this. 1, 2, 3, 4, 5, 6, 7. And it has one job. It functions simply to activate the G protein. The G protein is what we call a heterotrimeric protein. Tri meaning 3 different parts, hetero meaning those parts are all different. And we're going to divide this up into the alpha subunit and the beta gamma subunit. I draw the beta gamma subunit together because these won't separate, but the link between alpha and beta gamma is very, very important to understand. In this configuration, alpha is bound to a GDP. And in this scenario, the fact that alpha is bound to GDP allows beta gamma to essentially bind and tether alpha to keep it from doing its job. Think of a kid that's a little bit wild in a store and wants to go crazy, but the mom puts the kid on a leash. That's what's going on here. Beta gamma being the mom and alpha being the crazy child. If alpha gets loose, things are going to happen. So the job of the receptor simply is to help alpha transition from GDP to GTP, and we'll show you how that happens here in just a second. When a ligand binds, of course, a ligand is anything binds to a receptor. We've got lots of different GPCRs and lots of different ligands to bind. When a ligand binds, it causes a conformational change in this loop right here. Essentially, that loop's going to get a little bit bigger. It becomes more exposed, and now it provides a docking site for this alpha subunit. And as that happens and alpha comes and associates with this GPCR, the job of the receptor is simple. All we're going to do, here's a big pair of scissors, we're going to cut off the GDP, off goes the GDP, and on comes a nice new shiny GTP. And this is important because if alpha is bound to GTP, that allows alpha to liberate itself from the beta gamma subunit. And if alpha's loose, things are going to happen. There's a couple of different canonical signaling pathways now that we'd like to just talk about to highlight what might happen in any given scenario. One of the targets, now recognize I'm not going to have multiple targets in multiple different cell types. I'm only going to have one target in one cell type typically. One of those targets could be a membrane-bound protein called Adenylate Cyclase. And hopefully you can infer from the name what Adenylate Cyclase does. Its job is simple. Its job is to take ATP and convert it to a molecule called cyclic AMP. Now, if you're interested in how that happens, if we drew a quick ATP molecule, there's adenine, 2 hydrogen bonds, and then 3 phosphate groups. There's ATP, right? Well, if you remember, the phosphate group has a lot of oxygens on it that we're not drawing. Essentially, what we're doing with adenylate cyclase, we're going to cut off these phosphates here and actually create a bond, use that energy from the reaction to create a bond that links to one of the other oxygens on this first phosphate. And essentially what that does now is create a phosphodiester linkage here where this given phosphate is attached to adenosine right here on this carbon, the 5' carbon, and down here on this carbon, the 3' carbon. So 2 carbons from the sugar attached to one single phosphate, and that's why we call it cyclic. So this is cyclic adenosine monophosphate, or cyclic AMP. This is important because cyclic AMP is a potent second messenger. It does a lot of different things inside the cell. There's a couple of examples that we could show you. One of the things that it could do, it could bind, for example, to a kinase. So here we have protein kinase A. Along comes cyclic AMP, and when cyclic AMP binds to it, then protein kinase A is activated. And this could do lots of different things. It could phosphorylate ion channels. For example, maybe we had a sodium channel on the cell membrane. Once it's phosphorylated, that's going to open it up and it's going to let sodium in or calcium in. Those are just some examples of what protein kinase A could do. This entire process, if we go back to cell signaling, we have reception. All of this that we've got down here would be transduction. And then the activation here, whatever that does, would give us a response. There is one other GPCR signaling pathway that I would like to highlight, and it follows a very similar pattern to the one we just saw. We'll use some different colors to change it up. But the idea is the same. Here we have alpha tethered to beta gamma. These are of course both membrane bound, and this is bound to a GDP, allowing beta gamma to keep it in check. When a ligand comes and binds, that opens up a binding site here, allowing association with the alpha subunit and the G protein coupled receptor. And again, the job of the G protein coupled receptor is just one job. It's going to cut off this GDP and replace it with a shiny new GTP. And that liberates the alpha subunit. So here's alpha. It's bound to a GTP. And in this cell, the other target that we want to mention is a target called Phospholipase C. This belongs to a class of enzymes called phospholipases, and just as their name suggests, its job is to cut a phospholipid. If we were to zoom in on the membrane, of course this is a lipid bilayer, so we zoom in here and we have our outer leaflet, we have our inner leaflet, and then we have all of our fatty acids, right? Well, phospholipase C targets a phospholipid on the inner membrane called PIP2. So here's PIP2. You would recognize this. We're just going to draw it this way. 2 fatty acid chains, glycerol backbone, and then a phosphate group, and then a polar head group. And in this case, we're actually going to define this polar head group. It is a 6-carbon sugar that is phosphorylated here and here. This is phosphoinositol bisphosphate. The sugar that we just drew is the sugar inositol. Remember, all of this is just a phospholipid. It's one of these guys, right? It's a phospholipid inside the cell membrane. We've drawn it bigger to help you see what phospholipase C is going to do. So when phospholipase C is bound to the alpha subunit, and the alpha subunit is available because it's bound to a GTP, then the job of this enzyme is, again with the scissors, we're going to cut the phosphate group and the polar head group off of the glycerol backbone. That's actually going to give us two different molecules. If we extend the cell membrane down here, this is going to give us this molecule, 2 fatty acid chains and glycerol, so we call this DAG, which stands for diacylglycerol, or DAG, and this molecule here, which is IP3 inositol triphosphate. Inositol again is the ring structure, and you can see that it's bound to 3 different phosphate groups. IP3 in this scenario is our second messenger. And IP3 has a very special job. Once it's cut off of PIP2 at the cell membrane, its job is to come down here. Here we have the smooth ER. And if you recall, we know that through primary active transport, let's draw this. This is an ATP-driven pump, primary active transport. This is called the Circa pump, which stands for smooth ER calcium. And its job is to just use ATP and pump calcium into the smooth ER. And we talked about this previously. One of the functions of the smooth ER is to store calcium. This is how it does it. Well, now we can begin to see the target of IP3. So here in the smooth ER, we have a ligand-gated calcium channel, and that ligand, you guessed it, is IP3. So as IP3 comes down, this is IP3, as it comes down and binds to this ligand-gated channel, that's going to cause this door to swing open. And once it's open like this, that is going to allow calcium to come out of the cell. Of course, this is going to be facilitated diffusion, facilitated because calcium is charged and can't go through the membrane by itself, but diffusion because it's going down its concentration gradient from high to low. Now, calcium represents another second messenger. And depending on the cell system that we're in, this calcium could do lots of things. One of the things that it might do is come up and interact with, let's draw it like this. This is protein kinase C, or PKC. And protein kinase C is activated by 2 different signals, one of them being DAG. So as DAG comes over here and binds to protein kinase C, we also need another signal, and this is where calcium comes in. So calcium can come up and bind to protein kinase C, and that causes PKC to be released from the membrane, and now we've got an active kinase. So again, we see the idea of reception, transduction, response. And of course, the response is going to be dictated by whatever cell we're in. If we're in the heart, for example, it's going to be different. If we're in kidneys, whatever we're talking about, the response is going to be different. But this part of the signal transduction pathway associated with G protein coupled receptor signaling is always going to be consistent.